Good day to everyone joining us, and welcome to today's Eurofins Cerep-Panlabs webinar. Today's talk is entitled Epigenetic Profiling in Drug Discovery, and my name is Mike Futala, and I'll be your host today. Today's webinar will run for approximately 60 minutes and includes a question and answer session. So this webinar is designed to be interactive, so please feel free to submit your questions and comments for our speakers throughout the presentation using the question chat box. And we will attend to your comments and questions during the Q&A session. This chat box is located in the control panel on the right side of your screen, and if you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in a listen-only mode and the presentation slides will advance automatically. And also please note that this event will be recorded and made available to you at a future time at the Eurofins Panlabs website for future review. So now, a brief history about the company. Eurofins Cerep-Panlabs Incorporated is a global CRO specializing in discovery pharmacology testing services for over 40 years. Eurofins Cerep-Panlabs' mission is to provide pharmacological testing services that predict clinical effects. And Eurofins Panlabs' scientific portfolio consists of over 1,800 assays, ranging from molecular assays to cell-based models through proof of concept in vivo activity determination. So now I would like to introduce our speaker for today's event. He is Dr. Manoduth Ramnath. Dr. Ramnath is responsible for the development of new classes of catalog assays by taking into account the latest regulatory requirements, market needs, and recent scientific discoveries. He has supervised the implementation of the Epigenetic platform at Eurofins Cerep-Panlabs, and he is also adept at developing customer-specific assays which are not present in the commercial catalog. Dr. Ramnath's main area of focus is coordinating screening programs with pharmaceutical partners in the field of epigenetics. Dr. Ramnath received his PhD at in biochemistry from the University of Stellenbosch, South Africa. So now I'd like to hand over the presentation to Dr. Ramnath. Please remember to continue to chat in your questions in the chat box, and we will address them in the question and answer session which follows the presentation.
Thank you, Mike, for that introduction, and I'd also like to extend a warm welcome to everybody today to this webinar. As Mike has indicated, we'll be discussing epigenetic profiling in drug discovery. In today's talk, we will cover the following topics. The first is, very briefly, we'll go over what is epigenetics and what we consider to be important in assay development here at Eurofins Cerep-Panlabs, and how we've implemented that and how we continue to do this in our constant development and striving for the best quality assays that we can provide you with. We will then go through a case study of the development of a histone methyltransferase, namely a G9A. We will also cover protein production and data generation capacities here at Eurofins Cerep-Panlabs, and we'll also be covering a very new exciting technology platform that we just brought online, namely mass spectrometry. And finally, we'll end off with how we've empowered two of our very powerful tools that we already have here at Cerep-Panlabs, which is namely the Bioprint and ANCA Panel. So without further ado, the first question is, what is epigenetics? Epigenetics refers to molecular mechanisms that cause heritable changes in gene expression without altering the DNA sequence. It's important to realize that there are three main factors that contribute to the epigenetic regulation of gene expression. These are non-coding RNAs, DNA methylation, as well as histone modifications. The deregulation of epigenetic control is a common feature of a number of syndromes, disorders, and cancers. Epigenetic deregulation has been correlated to the following pathologies, namely inflammation, cancer, tumors, viral infection, metabolic, neurological, as well as immune disorders. This field of research is now considered to be a novel avenue of chemical safety assessment and drug discovery. Many of you who are new to this field, or just as a quick revision, many of you may have heard the three key words used often to describe the players in the epigenome or the epigenetic code, namely the readers, erasers, as well as the writers. Very briefly, a writer is an enzyme that will add a chemical moiety, either to the histone or, as indicated in my list, also to DNA, for example the DNA methyltransferase. Erasers are simply enzymes that can remove the chemical moieties. A very quick example would be histone demethylase. And readers, on the other hand, would be proteins that bind to chemical moieties. To give you a more full story, for example, with the histone, for the histone acyl marks, a histone acyl transferase will add on an acyl mark onto the histone tail. An eraser, notably the hashDAC or certwin, can remove this particular acyl mark, and a reader, which will be the bromodomain in this case, will be able to detect this particular mark. We would like to demonstrate how we can help you in your epigenetic inhibitor discovery program by using our current offer, which includes our catalog assays, how and why we have chosen to produce our own proteins here on site. This allows us then, with our capacity to master several technology platforms, to be able to offer custom assays. And finally, as indicated earlier, I will finish off with how we have empowered both our Bioprint and ANCA Panel tools to be able to detect overall promiscuity of lead compounds as well as determine predictive biomarkers. Today, Eurofins Cerep-Panlabs has the largest commercial portfolio of targets in the epigenetic arena. We have over 55 writers. These include kinases, DNA methyltransferases, histone methyltransferases, as well as histone acyl transferases. You may notice that we also have small molecule methyltransferases, or as many people tend to refer to them as SMMTs, which are developed in the mass spectrometry format. I will cover that later on in this webinar. We also possess 47 different readers, and we very recently added 19 methyl readers to our catalog. We also have the traditional erasers, as well as cell-based assays, which are able to detect specific epigenetic modifications on histones within cells. Today, we have 162 assays in epigenetics which are available right now, and you can actually get this information from our website at Eurofins Cerep-Panlabs. This brings us to a very important portion of this webinar, namely the assay development. Several of you who actually set up in vitro assays in the lab are often confronted with a very important question. What makes a good assay? For us, we always strive to ensure that we are able to cover the four main criteria. Firstly, being the purity of the protein. If it's an enzyme, we really are very careful to ensure there's a high specific activity. We also ensure that the substrate that is used is biologically relevant. We also require to have a reliable technology platform to be able to monitor, in the case of an enzyme, the kinetics of the reaction. Finally, we also strive to set up all our enzymatic reactions in the initial rate period, which is the most biologically relevant area to be in for an enzymatic inhibition reaction. Going further in depth into our assay development process, for today's webinar, I'll be talking to you about how we go into the development for a writer assay. Initially, we look at the literature to identify the appropriate substrates and cofactors which have been associated with the particular writer. However, in epigenetics, it's often that different researchers use various substrates, and which is reported in the literature. This is why we always verify the substrate specificity here in our lab. For example, we will test systematically for each writer the nucleosome, core histone, full-length histone, and if possible, a specific peptide. Based upon the signal-to-noise ratio, we will then choose the most appropriate substrate. We will then move on to the next step, which involves the optimization of the reaction conditions. It is very well known that there could be interfering properties with certain types of ions and surfactants. These are all taken into account, and once we obtain the best signal-to-noise ratio, we maintain these conditions and determine the apparent Km of the substrate and the cofactor. I would like to point out, and it's very important to note, that we set up our enzymatic reactions at the Km for both the substrate and the cofactor, and we maintain these conditions up to and including the industrialization step, or in other words, the assay that you will be able to purchase from us. Another very important point is that we are in the initial rate period, which is also maintained in the assays, in our catalog assays. Finally, in the industrialization process, we also verify plate uniformity and the stability of our reactives. For our writer assays, all of them are based on the gold standard radioactive platform, which is very well known to be highly sensitive and to have a large dynamic range, which allows one to adjust simply the substrate concentrations. Radioactivity is also compatible with several different substrates: the nucleosome, core histone, full-length histone, and peptide. But very importantly, no substrate labeling is required. Neither does one require to perform a coupled reaction, enzymatic reaction, or neither is one required to use antibodies, which is a big plus. Just to give you a quick example of how we perform this, I'll be showing you in this particular slide how we performed the assay development for the PRMT7 histone methyltransferase. We initially determined the Km of the substrate as well as that of the cofactor. At the bottom left-hand corner, you can observe the kinetics of the final reaction. It's important to note that the blue filled squares are those of the actual kinetics of the reaction, with the black filled squares being those of the background. The arrow indicates the exact point at which we stop the reaction. This is the actual conditions in which we stop the reaction in our catalog assays, indicating that we are well within the linear range of this particular enzyme and the other enzymes in our catalog. We also determine the inhibition curves for each enzyme, notably here for PRMT7, using different well-known and characterized inhibitors. And we always try to ensure that we are well within the reported inhibition levels indicated for the particular enzyme. We will now move on to the case study using the histone methyltransferase G9A. It's very often when we start setting up an in vitro assay, we often ask ourselves a very simple question: which is the best substrate to use? And furthermore, does the IC50 value change due to the substrate used? In order to respond to this question, we have used, as indicated, G9A, which is a histone methyltransferase, using the radioactive filter-based assay. For this particular experiment, we tried all 4 possible substrates for G9A, namely the nucleosome, a core histone, full-length histone H3, as well as a peptide. We were able to observe that there was no significant differences for universal inhibitors of SAM, namely the SAH, for example. However, we did notice that there were significant differences in the IC50 values for specific inhibitors, for example, the BIX and the UNCO 638. Incidentally, we were quite surprised to notice that a non-specific inhibitor was also found to inhibit G9A, which in this case was the COMT inhibitor 41096. We took our hypotheses one step further. Apart from just about which does the substrate change IC50s, we also asked a very simple question: does the technology platform used for the detection of a signal alter or modify the IC50? For this particular experiment, we could not use all types of substrates due to different technology platforms. We were use the peptide H3 1 to 21. Once again, we used a filtration radioactive based assay, an SPA assay which is also radioactive based, the Lance, as well as alpha screen assays. Once again, we observed no significant difference for universal inhibitors, but we did observe significant differences for the two specific inhibitors that were studied. It's also interesting to notice that the SNR that we obtained in all of the different technology platforms were very similar. In conclusion, we were able to produce 4 HTS adaptable technology platforms which were optimized using 4 different substrates. Certain inhibitors were detected with a higher affinity using a peptide substrate in comparison to other biological relevant substrates, namely the nucleosome or core histone or the full-length histone. Radioactivity filter binding assays allow for the rapid substitution of epigenetic substrates. Due to the fact that we were beginning to observe a COMT or a non-specific methyltransferase inhibitor inhibiting histone methyltransferase, we believe that histone methyltransferase drugs should be screened against an off-target panel as well. Moving on to protein production and data generation capacities here at Eurofins Cerep-Panlabs. When we started this particular platform in epigenetics, we were rather surprised at the quality of proteins that was commercially available. This is why we took the bold step of being able to produce our own proteins in-house. For example, in 2013, we produced over 130 proteins. It's interesting to note that we have the capacities to produce proteins from either E. coli or from eukaryotic cells, namely the SF9 insect cell lines. We have a dedicated team that's able to optimize and produce proteins, and we always ensure that we maintain 4 levels of internal quality control. The first being, we always ensure that we have the fully sequenced plasmid and insert, as well as we constantly monitor each batch of protein produced, and we have historic data that traces each batch that's produced. And if we find there's more than a 30% difference in activity, that particular batch of proteins is invalidated immediately. Finally, we also determine via mass spectrometry the identification of each protein. The reason why we do this is to ensure that there's no degradation of the protein of interest, as well as we like to monitor if there's any post-translation modifications that has occurred to the protein which could modify its biological activity. Our capacities to produce proteins does provide new opportunities to go from gene to protein. As many of you may know, several proteins of interest are not commercially available. Our protein production capacities will also allow one to produce truncated proteins if one is interested to look at only the active site or target an alternative site on the same protein. We are also able to clone and produce mutated proteins, as several diseases are due to mutated proteins which are often not commercially available. We have also been able to co-express certain number of histone methyltransferases with transcriptional factors which are known to modify their enzymology as well as the types of inhibitors which are able to inhibit well-known histone methyltransferases. All our assays in our catalog are HTS or high throughput screening compatible. In order to perform these, we have a dedicated team in the compound management. For example, we have several liquid handling robots, as well as in 2013 we were able to prepare, for both screening and profiling purposes, more than 2 million compounds. Apart from compound handling, we also have a specialized team that's able to perform the actual screening itself, and we are able to choose a particular team depending on the technology platform on which the particular HTS is based. For example, we are able to produce up to 90,000 data points per day for an enzymatic assay. Based on the fact that we are able, and we do, master several technology platforms, coupled with the fact that we can modify proteins and enzymes, as well as change substrates for a particular assay, we are able to modify an entire assay which could be required for medium and high throughput screening, profiling, lead optimization, as well as off-target effects. Once a particular hit has been determined, we can also determine the actual type of inhibitor using classical enzymology, which will give us more information about the type of inhibitor, namely is it a competitive or non-competitive inhibitor, a mixed inhibitor, as well as the reversibility of the particular inhibitor of interest. We can also perform orthogonal screening on a particular platform, either using surface plasmon resonance or SPR, thermal shift, or mass spectrometry. And with that, I'd like to move on to our latest technology platform, namely mass spectrometry. Just to give you some of the main advantages of mass spectrometry and why we have decided to adopt this particular platform in epigenetics, is that one does not require any chemical labeling. It does not require, it allows one to look at the direct measurement of multiple reaction products concurrently. We're able to reduce the method development time. It allows us to perform label-free analysis of native compounds for functional biochemical assays. Furthermore, we can also look at multiple epigenetic marks simultaneously. And due to the type of automated injector that we have, we can produce a data point every 6 to 10 seconds. To give you an example as to why we say we can reduce the method development time, we have adopted a universal approach for the detection of SAM methyltransferases, in which we are able to monitor the product from the co-substrate, namely SAM, or we are able to monitor the product, namely SAH, which is produced from the SAM once the SAM has donated its methyl group onto the substrate, which could be the histones or small molecules. The case study I'm going to show you today in today's webinar is that of a small molecule methyltransferase, nicotinamide N-methyltransferase, or NNMT. Even though we have opted to look at the production of the SAH, we are completely capable of being able to observe the substrate, namely nicotinamide, as well as the methylated product. We are also able to observe a reduction in the cofactor, the SAM, and an increase in the levels of the SAH. In this particular slide, we are simply looking at the substrate Km determination, in which we are monitoring the cofactor product SAH, which increases as a function as an increase of concentration of the nicotinamide substrate. As indicated earlier, we always ensure that we produce our assays in the best enzymatic conditions possible. And once again, even in our mass spectrometry platform, we determine the Km values both of the substrate and of the cofactor. Here we periodically, and not only for the mass spectrometry platform, but our other enzymatic assays as well, we determine the DMSO tolerance as well. For this particular case, it is 8.9%. It has an IC50 at 8.9%. You may have noticed that we have used the product as an inhibitor for this particular assay. This is due to the fact that we could not find an inhibitor for this particular small molecule methyltransferase, a specific inhibitor. However, in order to validate our assays, we always like to ensure that we can inhibit the enzymatic reaction, and for this particular case we had no choice. This is why it will be up to you, the listeners, to find specific inhibitors using our test in the future. Incidentally, we also have used the non-specific inhibitor SAH to be able to inhibit this particular reaction as well. For the moment, we have 7 assays that make up this panel of SAM-dependent methyltransferases, which can aid you in determining if your inhibitors for the histone methyltransferases are specific or not. Having enabled this particular platform and set up these assays, we simply used very specific histone methyltransferase probes from our partners, the SGC, and screened them across our panel. We were quite surprised to find that small molecule methyltransferases are inhibited in the micromolar range. And I would like to draw your attention to the UNC 0642 and the UNC 0638, which are specific G9A inhibitors and which normally have been reported to function in the nanomolar range. However, we find that it's inhibiting the small molecule methyltransferases in the micromolar range. So there's a few logs difference, and this shouldn't be a problem whatsoever. We were rather surprised by the fact that the arginine methyltransferase inhibitors, namely AMI1 and egalic acid, which are known not to compete with the SAM binding pocket, were found to have almost no effect on most of the small molecule methyltransferases. On the other hand, BIX, which is reported to have activity in the micromolar range against G9A, was also found to hit non-specifically the small molecule methyltransferases in the upper micromolar range. So this could be an indication of a type of molecule that could be considered to be slightly non-specific. I would like to now share with you how we can observe multiple products on mass spectrometry. And in order to illustrate this, I'll be sharing with you the example of JMJD2, which is a histone histone demethylase, which is an eraser, part of the eraser group, which is known to remove methyl groups from the histone peptides. Here on the top we have the histone peptide H3 with a lysine, with a trimethylated lysine, and in the lower half of the slide, after the action of the demethylase, we can observe clearly three different subpopulations. Firstly, the remainder of the non-demethylated peptide, as well as the products of the methylated peptide, namely the dimethyl and a monomethyl population. The capacity to be able to monitor all the different populations of peptides in a particular reaction allows one to be able to generate IC50 inhibition curves on the product of interest. On this particular slide, we were monitoring the appearance of the dimethyl population. Normally, this would be done with an antibody. And if we wanted to monitor the monomethyl, the generation of the monomethyl population, we had to use a second antibody. However, using mass spectrometry, we can observe the two in a single reaction. The next case study is that of a writer, which is a protein arginine methyltransferase, or PRMT7. This is a very simple case in which we were using the substrate, a peptide substrate, and on top we have the spectrum associated with just the peptide alone, and at the bottom we have the spectrum after the action of the enzyme. And here on the lower right hand corner we can observe the population of peptides which have been monomethylated. In this particular slide, we are able to observe the inhibitory effects of the non-specific inhibitor SAH on this particular histone methyltransferase, and in this particular case we are monitoring the appearance of the monomethylated peptide. Finally, the last case study I'd like to share with you today in terms of the mass spectrometry platform is that of a full-length histone. And for this particular case, the writer used, or which is the histone methyltransferase G9A, we were able to observe on the top an unmethylated full-length histone as well as an oxidized state. Now this oxidized state is due to the methionines in the histone which are oxidized due to the fact that the histone is placed in an aqueous solution. In the lower half of this slide, we have the product once we have the action of the full length once we have the action of the histone methyltransferase, in which we are able to discern once again the oxidized state, unmethylated full-length histone, as well as the three populations of mono, di, and trimethyl full-length histone. Take into consideration the fact that we have on-site protein production capacities as well as various technology platforms. Eurofins Cerep-Panlabs can provide a unique service from gene to data. This means that we are able to clone a protein of interest, produce the protein, set up either an enzymology or binding assay, perform the reaction, and produce a readout using different technology platforms, namely mass spectrometry, radioactivity, high content analysis, qPCR, or TRF rate. We can estimate that the average turnaround time for a project like this will take about 3 months. Eurofins Cerep-Panlabs has always been committed to the de-risking of compounds. This is why we took the incentive to answer the question, should epigenetic targets be a concern in safety pharmacology assessments? This has been an internal effort from Eurofins Cerep-Panlabs in which we have screened over 1,000 FDA compounds, approved compounds, on over 16 epigenetic targets. Now, in order for me to be able to explain how this was done, I first need to cover a well-known homogeneous database that is present here, which has been put into place at Eurofins Cerep-Panlabs from 1997, namely Bioprint. Now, Bioprint is the first comprehensive homogeneous database to link both chemical and in vitro biological descriptors to clinical properties. So how does this work? We have compounds, and I will list that in my next slide, in which we have measured data on in vitro targets, and as well in my next slide I will indicate what these in vitro targets are. It's very important to realize that all the measured data for this particular data set has been performed here at Eurofins Cerep-Panlabs. This information was not correlated simply from literature, so we can perform the tests after several years using the same SOPs and get exactly the same results. More importantly, the in vitro targets have been, by using predictive associations, been associated with in vivo effects. The biological fingerprints have been described as an alternative means to describe compounds of biological interest. But the combination of both biological fingerprints with the structure-based descriptives are more powerful than either one alone. And this allows for in vitro, in vivo relationships such as adverse drug reactions. I would like to point out at this stage that the molecules that we have in our Bioprint database is composed of more than 2,500 compounds, 60% of which are marketed drugs. I'd like to point out that we have 2% of these drugs which were withdrawn from the market, 20% of standard pharmacological references, and 12% of other compounds. Now, these other compounds are degradative compounds that are formed once a metabolic compounds that are formed once a particular molecule of interest is being degraded. These are then pitted against 159 in vitro assays, which include receptors, enzymes, ion channels, transporters, as well as ADME assays. These in vitro assays were then correlated to in vivo effects from data in the literature. These include therapeutic classes, adverse drug reaction profiles, PK profiles, drug-drug interactions, as well as mechanism of action studies. The Bioprint database has been used by our pharma partners for the identification of promiscuous compounds as well as for the detection of adverse drug reactions. Just to give you a quick idea of how one could actually use Bioprint will be as follows. The new candidate can be run on the same assays as the Bioprint compound. The profile will then be analyzed. Each hit can be analyzed for an adverse drug reaction, or alternatively, as indicated in the right-hand side panel, the entire profile can be used to identify compounds with similar profiles. In this particular example, we did not even have the chemical structure associated with the compound of interest. However, using the biological footprint left behind by this particular molecule with respect to its activity against the different assays in the Bioprint, we were able to find the nearest neighbors to this particular seed profile. And using the nearest neighbors, one can either reposition a molecule in terms of what is its probable activity in relation to known molecules on the market, or alternatively, if your seed profile is very close to a particular neighbor with a known adverse drug reaction, this could indicate that your molecule of interest potentially could have similar adverse drug reactions. In a nutshell, Bioprint allows one to position new drug candidates in context of drugs and related compounds currently on the market. Not only did we want to understand if epigenetic targets should be of safety concern, we also wanted to determine if epigenetic targeted molecules could have effect on housekeeping proteins, resulting in adverse drug reactions. As you may recall in my previous slides, Bioprint did not contain any epigenetic targets. This is why we added in a total of 16 of them. These included 5 writers, namely the histone methyltransferases, 5 demethylases, 3 HashDACs, 3 bromodomains, and 1 DNA methyltransferase. Indicated in this slide is a heat map of the epigenetic data set, with on the y-axis the names of each of the epigenetic targets studied, and on the x-axis from left to right the 1,000 FDA approved drugs that have been run against the 16 epigenetic targets. Red indicates the most active compounds, going all the way down to green indicating that they were inactive. And what you'll be able to observe from this is that marketed drugs do interfere with epigenetic targets. As a case study, I'd like to zoom in onto this particular antimicrobial interference that we were able to detect. We were able to observe that an antifungal as well as a third generation antibiotic were found to interfere with several of our epigenetic targets that were tested. I would like to draw your attention to compound number 2, which on the Bioprint profile was found to have almost no hits at all, indicating that it has almost no known adverse drug reactions, or either could not be detected using Bioprint. However, this particular compound was found to hit 9 out of the 16 different epigenetic targets that were tested for the epigenetic profile. In conclusion, certain classes of commonly used pharmacological compounds do interfere with epigenetic targets. Current and new compounds should be screened for the perturbation of epigenetic targets for safety reasons. Bioprint can be used to flag molecules in the early stages of development, because not only you will be able to have a look at the epigenetic data set, but as well as the overall promiscuity of the molecule. This data set can be used for the determination and identification of chemotypes that interact with individual epigenetic targets, as well as later on, and we are currently looking into the use of the data set for designing epigenetic target panels for safety assessment. I must admit, we were rather surprised by our find, and this is why we wanted to really confirm the nature of these hits, and this is why we followed them up in a cell-based assay using the cell-based assays that we have in our catalog. We used 4 cell types, and we exposed these 4 cell types to the antimicrobial compounds that were detected from Bioprint. For this particular case study, we incubated them with an exposure time of 24 hours to the antimicrobial compounds. However, this can be extended right up to 72 hours. We normalized the data to the ATP and viability, as well as to the full-length histone H3. Even though we looked at several epigenetic targets or marks, we were able, and what I have on this particular slide is the histone H3 lysine 9 dimethylated. On the other marks, we had very small changes, below 20%. That is why I am not showing those to you. But it's interesting to notice that these molecules were able to have cell-based effects, and the effects on each were found to be cell line dependent, which can be observed as a function of the inhibition rates of this particular target on the different cell lines. However, working in the oncology field, one may like to monitor the phenotypic effects on several cell lines and not just a simple model that I just shown you. And this brings me to the ANCA Panel. Now, the ANCA Panel is a much more complicated and complex system which involves 240 cell lines for which we have extensive genomic characterization. We have the full mRNA expression levels, mutation and DNA copy number data, which are known for each of the cell lines which comprise the ANCA Panel. You may notice that we have very recently enabled ANCA Panel with the 3D tumor spheroid capacity. This is of extreme interest to epigenetics due to the fact that there's quite a few publications and literature that's coming out now that indicates that the susceptibility of cells in 3D models in comparison to 2D models do change. Additionally, we have also re-optimized this particular panel of assays for slower acting drugs, which is often the case for epigenetic drugs, which require prolonged exposure. The ANCA Panel has now been validated for assays up to 10 days. However, it's highly customizable, which indicates that you can stop your assay after 3, 5, 6, or up to 10 days. For the ANCA Panel, one can run small molecules, biologics, and extremely important for epigenetics as well, one can also look at drug-drug combinations. After exposure of your cells to your molecules of interest, high content analysis is performed, and we standardly monitor cell count, apoptosis, as well as the cell cycle. However, custom readouts can also be performed. Taking into consideration the IC50, the genomic data, as well as histology, one is able to determine the mode of action as well as identify and prioritize predictive biomarkers. For today's talk, I'll be sharing with you some of the data that we have obtained using the GSK343 inhibitor, which is a histone methyltransferase inhibitor of the EZH2. On the top left-hand corner, we have the effect of the GSK343 on the Pfeiffer cell line. In blue, you have the cell count after a 3-day exposure, and in pink, the 10-day. Below that, on the same cell line, you have the effect of UNC 1999. And very interestingly, you can note as well that there is no shift for the pan-kinase staurosporin control. However, for both GSK 343 as well as UNC 1999, we can observe a left shift. This is surmised in the bar graph to the right, in which we have the ratio of the 3-day to the 10-day IC50 values, in which, for example, for the RL cell line, we can observe up to a 300-fold difference in activity. Incidentally, below, you are able to observe a custom readout, in this case being the monitoring of histone H3 lysine 27 trimethylated. Using the ANCA panel, one is able to correlate sensitive and resistant cell lines to specific and pathway biomarkers. It also allows one to investigate multiple types of cancers and multiple mutations in order to determine specificity and selectivity. With that, I would like to conclude, and I hope I've been able to show you why we should come to Eurofins Cerep-Panlabs for your epigenetic program. Not only do we have the largest commercial catalog, and that all our assays are MTS and HTS compatible, we also have more than 2,000 other assays in our catalogs which are required for the further characterization of your molecules of interest. And please do not forget that we have over 250 in vivo models as well. So consider us as a single company for all your epigenetic services. And with that, I would like to conclude and thank you for your attention, and I would now like to take a few questions.
Great. Thank you, Dr. Ramnath, for that very interesting and informative presentation. So we'll now begin the Q&A session, and I'd like to invite the audience to continue sending their questions and comments right now using that chat window. So we've received a few questions. We have time for a couple. I'll remind the audience that we'll compile all the questions and send the answers after the webinar, but we can do about 2 questions. We will take about a minute to review, and then we will be back momentarily. We appreciate your patience. Thank you.
Thank you, listeners, for your questions. We'll start off with the one that we have, which is, are you able to produce protein complexes? Now, this is a good question, and as we very well know that most enzymes do not only have a catalytic area, but also they have several other enzymes that interact with them in order to give them their full activity. And the only example that I have to be able to give you that in which we produce on-site protein complexes will be that of the MLL complex. If my memory serves me right, our MLL complex consists of 3 components: the WR5, the RBB5, and the MLL portion. We also have PRMT5 and PRC2. The next question that we have is, what are the antimicrobial compounds that were referred to in the Bioprint? Now that's a very good question. Unfortunately, I cannot give you those answers directly. Bioprint dataset, as indicated, was an internal effort. So for the moment, it's still a pay-per-view dataset. The information in this dataset is commercially available either by target or by molecule. So please do not hesitate to contact one of our representatives, business directors, to be able to determine the pricing that's associated with that. Incidentally, this is just one example, one case study of antimicrobial compounds that we found. We've also found several other very interesting correlations. Another question that has just come through is, are your proteins commercially available? Our response to that is yes, they are. We can supply you with the proteins that are in our catalog. Alternatively, as indicated in the webinar, we can also customize proteins for you, either looking at mutated proteins, full-length proteins, truncated proteins, and so forth. So once again, for this particular service, do not hesitate to contact our commercial sales force.
For this event. A survey window will be popping up on your screen, and your presentation is appreciated as it will help improve our future webinars. Now please join us in thanking our speaker, Dr. Ramnath, and we hope you found this conference informative. Please have a great day, everyone. Thank you.